triiodothyronine, which is known as T3, and its prohormone thyroxine, which is also known as T4, make up the thyroid hormone. Both are tyrosine-based molecules which is conjugated to iodide. 3 iodides for T3 and 4 for T4. T4 is inactive while T3 is the active form. T4 is converted to active T3 form by the deiodinases. In this lesson, we will focus on the synthesis of T3 and T4, which occurs in the thyroid gland in a multi-step fashion. Let's begin on how the cells from the thyroid gland take up iodide from the plasma. It starts from the transfer of iodide ions from the plasma into the follicular cells across the basolateral membrane via the sodium iodide cotransporter. It uses the sodium electrical chemical potential to drive the transport of 1 iodide ion and 2 sodium ions. Once inside the cell, the extra sodium ions are transported back out of the cell via the sodium-potassium ATPase pump, while the iodide ions is transported across the apical membrane into the colloid space via the pendrine cotransporter. Pendrine transports 1 iodide ion into the colloid space in exchange for 1 chloride. Once inside the colloid space, the enzyme thyroperoxidase oxidizes 2 iodide ions to form 1 iodine molecule. Next, the peroxidase iodinates the tyrosyl residues of thyroglobulin protein. Iodinating a single tyrosyl residue forms monoiodotyrosine, while iodinating 2 tyrosyl residues forms diiodotyrosine. Iodination of tyrosyl residues causes the thyroperoxidase to conjugate neighboring tyrosyl residues. This is important to know because it results into 3 different types: T4, T3, and RT3. RT3 is biologically inactive. The thyroglobulin protein involved in this process was initially synthesized in the endoplasmic reticulum of the follicular cells, secreted into the colloid space via exocytosis. Following the iodination and conjugation, the iodinated thyroglobulin binds megalin, which is an endocytic receptor located in the apical membrane of the follicular cells. Binding in megalin promotes the endocytosis of the iodinated thyroglobulin, after which lysosomes containing proteases fuse to the thyroglobulin containing vesicle. Proteases then cleave the peptide bonds that flank where the T4, T3, or RT3 were located on the peptide backbone of the thyroglobulin. Once cleaved, T4 and T3 are transported out of the cell across the basolateral membrane via the monocarboxylate transporter.
Created using Powtoon.